In this video tutorial, I am going to discuss beta oxidation of unsaturated fatty acids. Beta oxidation of unsaturated fatty acids is very similar to that of saturated fatty acids except for a couple of key differences. To clearly understand what makes it different, let's look at a real example. So let's take linoleic acid as an example of unsaturated fatty acid, and this is the structure of linoleic acid. As you can see, it has 18 carbons and 2 double bonds. Now to describe the position of these double bonds, we use the delta numbering system. In this system, carbons are counted starting from the carboxyl group, and it is shown here in the red color. The first double bond is between carbon 9 and 10. We use lower numbers, so this double bond is delta 9. The second double bond is between carbon 12 and 13. That's delta 12 double bond. These both double bonds are in cis configuration. Now, beta oxidation works by removing 2 carbons at a time, releasing them as acetyl-CoA. Let's see how this happens. In the first cycle, this bond is cleaved and one acetyl coenzyme A is released. In the second cycle, the same thing happens. Third cycle, again the same thing. Notice that these first 3 cycles are completely normal. They are exactly same as saturated fatty acid oxidation, nothing special here. But now in the fourth cycle, things gets interesting. We need to cleave this bond here, but this double bond becomes problematic because it does not allow the beta oxidation to proceed normally. Originally, this double bond was delta 9, but we have already removed these 6 carbons. So now we need to renumber the carbons. This becomes carbon 1, carbon 2, carbon 3 and carbon 4. So as you can see, this double bond is now at delta 3 position and it is still in cis configuration. So this problem is called as delta 3-cis problem. So how does our body fixes this delta 3-cis problem? The double bond is shifted from between carbon 3 and 4 to between carbon 2 and 3. And during this shift, the configuration changes from cis to trans. In other words, delta 3-cis becomes delta 2 trans. This is an isomerization reaction. Nothing gets added, nothing gets removed, just a rearrangement of atoms. A specific enzyme called an isomerase carries out this reaction. Now here is the clever part. Normal beta oxidation has 4 steps, right? The step 1 actually creates a double bond at the delta 2 position. But we already have that double bond due to isomerization. So step 1 is skipped entirely and we directly proceed to step 2, 3 and 4 of beta oxidation. But this skipping of step 1 has its own consequences. Because first step normally generates FADH₂, which goes into the electron transport chain and produces ATP. Since we are skipping it, we lose that FADH₂, which means we lose ATP production. This means beta oxidation of unsaturated fatty acids generate a bit less ATP compared to saturated fatty acids. With the isomerization completes, the fourth cycle finishes and releases another acetyl-CoA. Now we can move on to the fifth cycle, where these bonds needs to be cleaved. And once again, we have a double bond which is problematic. Although it appears far from the cleavage site, it still interferes with the enzymes of beta oxidation. So what is the position of this double bond? By now we have removed these 8 carbons in total. So let's renumber again from this new end. So this becomes carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. After renumbering, this double bond lies between carbon 4 and 5. This is delta 4 double bond and again in the cis configuration. So this problem is called as delta 4-cis problem. This particular problem and its solution are little bit complex. So here I am intentionally simplifying the mechanism to highlight the metabolic logic rather than the molecular complexity. So how does the body solves this delta 4-cis problem? The simplified explanation is straightforward. Just get rid of the double bond completely by saturating it. The enzyme system adds one hydrogen atom to this carbon and another hydrogen atom to this carbon. This converts the double bond into a single bond. In other words, delta 4-cis is converted into a single bond. Where do these hydrogen atoms come from? They are donated by NADPH plus H plus, which gets converted to NADP plus. This is a reduction reaction and it is catalyzed by a specific reductase enzyme. Now notice the contradiction over here. We are talking about oxidation, a process that releases energy, but we just used a reduction step which consumes energy. You might be thinking that I don't see ATP being used directly here. That's true. But here is the thing. Regenerating NADPH from NADP plus requires ATP. So indirectly this step costs energy. So this gives us a second reason that why unsaturated fatty acid oxidation yields less ATP than the saturated fatty acid oxidation. First, we lose FADH₂ production where we skip step 1, and second, we consume ATP to regenerate NADPH during the reduction step. Once this reduction is complete and the Δ4-cis problem is solved, the fifth cycle of beta oxidation proceeds and this bond is cleaved. And now, as there are no more double bonds, rest of the beta oxidation cycle continues normally until the entire fatty acid is broken down into acetyl coenzyme A units. So let's summarize. During beta oxidation of unsaturated fatty acids, we run into two problems and both caused by double bonds. The problem number 1, the Δ3-cis problem. This is solved by a specific isomerase which shifts the double bond and convert it to Δ2 trans double bond. Problem number 2, the Δ4-cis problem. This is solved by specific reductase enzyme which saturates the double bond using hydrogen from NADPH. With just these two modifications, beta oxidation of unsaturated fatty acids proceeds almost the same as saturated fatty acids, just with a slightly lower ATP production. And that's all about the beta oxidation of unsaturated fatty acids. I hope that all the concepts are clear. If you have any doubts or questions, please write them in the comment section below. Thank you.